I am a scientific engineering associate at the Department of Energy's Joint Genome Institute, part of the Berkeley National Laboratory. And today, with the help of my son Nicholas, I'm going to explain to you how single-cell genomics works using props from my house because I'm under shelter in place. So let's say I have some dirt from my garden and I want to sequence all the bacteria in here to know what bacteria live in my dirt. But the problem is they're all mixed together, so I can't tell them apart, and they're in there with a lot of dirt. So the first thing I'm going to do is pour some water in and give it to Nicholas to shake and shake up. Can you shake this, Nicholas? Good job. Okay, now hopefully most of the bacteria are in the water, and so we're going to pour this through a sieve. And I have some dirty yard water that has bacteria in it. Step one accomplished. The next thing I'm going to do is I'm going to put a fluorescent dye in, symbolized by this food coloring. And Nicholas is going to stir it with a straw. Mix it up so the dye's in there, Nicholas. Alright, so now I have my cells in a solution. It's the water, but it's also other dirt particles. And I've added this dye. It's a fluorescent dye, which means that if I hit it with a photon of light, like from a laser, it's going to emit light in other colors that I can then detect with the detector when I run it through my machine. Importantly, the dye sticks to DNA. So only things that have DNA, like cells, are going to be bright, and things that don't have DNA, like dirt, are not going to be bright. For the purposes of this demonstration, Nicholas and I are going to be my flow cytometer. So we're going to take this water, suck it up through a tiny straw, and then all of the particles, the dirt and the cells, are going to go through the laser one at a time in a very small stream. This bucket of balls is now my dirt and cells. I'm going to hand them to Nicholas, who's the laser. He's going to hit them with light. If they light up, he's going to put them in the tray. If they don't light up, he's going to put them in the trash. Okay, Nicholas, hit it. Yeah, it has light. Put it in the tray. Okay, try hitting this one. Oh, no light, trash. Okay, try this one. Oh, no light, trash. Try hitting this one. Light into the tray. Okay, so we would do that for this entire bowl. And at the end, I now have taken my cells that were all mixed together and I'll have put them individually in a plate that has little reaction wells in it. So now I've completed the first step. Thank you very much, Lab Assistant Nicholas. Now that we have our cells in our plate, I'm going to talk about the other half of the single-cell genomics process at the Joint Genome Institute, which is the amplification of the cells. Let me turn my camera around. So I'm going to pretend that this sentence here is the DNA of one of the bacteria from my yard, and I want to read it. But the process of genomic sequencing where we read the DNA involves cutting the DNA up into little pieces, and then you only read really the middle. So if I tried to read one copy of DNA, it's likely to look like that, which makes no sense. So what I have to do is go from the one copy of DNA in my one bacteria to lots of copies. The way we're going to get from this one copy of my bacteria's DNA to lots of copies is called whole genome amplification. Setting this up is a lot like setting up a polymerase chain reaction, or PCR, where you have primers that attach to the DNA and an enzyme called a polymerase that's going to make copies of DNA. But in our case, we're going to use random primers that attach all over the entire genome and a special polymerase that is just going to make copy after copy for hours. So for example, if we had a primer at the M, the enzyme over time would just keep copying until it copied the entire sentence. If you have a primer at the I, it would start there and keep copying. And at the end of 10 or 15 hours, we end up with millions and millions of copies of our genome. What this means is that when we cut these up and we read just the middle of our cuts, we can end up with something like a puzzle that can be reassembled to make the whole readable sentence again at the end. I'm learning, my name is azobacter. I like to fix nitrogen. So that's basically my job, and I thought now maybe I would talk to you about how I got there. I got interested in science as a young girl because I really loved marine biology. I love dolphins and whales and the ocean. I scuba dive, and I started reading a lot of books about that. But when it came time for me to go to college, I decided to study agriculture and the molecular aspects of agriculture, like plant breeding, because I thought helping to grow food would be a really great way to sort of do something meaningful. When it came time for me to graduate, I graduated out of UC Davis with a master's degree, and I went to go work in a lab. And what it turned out I loved to do was hands-on lab stuff. So not so much asking big questions, but working out the small technical details to help someone else answer those questions correctly. And that really segued well into my work at the JGI. The Joint Genome Institute is a user facility, which means we've concentrated all this very expensive equipment and really hard to get training in one place. So lots of people from around the world who want information on their samples or their experiments but don't have the money or resources to do those kind of experiments can just come to us and they fill out an application and then we do it for them. I really love this idea. I feel like we help everyone. I feel like it's very collaborative. And I think it's also just very efficient. It wouldn't make sense for everyone to reinvent the wheel. It makes much more sense for everyone to come to one central place to do this work. In the future, the experiments I'm working on right now are to get more information during that sorting phase that my son demoed for you. So instead of just this binary, does it have DNA, is it a cell, let's analyze it, to put in other dyes. So things that might ask the question, is this an active cell or an inactive cell? Is it mostly just asleep in the environment, or is it doing something interesting? Or maybe you might have a cell you're interested in, but it's only 1 in 1,000 cells. Well, maybe we can put a label in there that will only attach to that cell, and so we can pull it out of the community. That's where my work is going. Outside of work, I love to bake, and I love sci-fi and fantasy novels, and a lot of my time is taken up now with my 2-year-old. One of the things I love about my STEM degree is that it helps me outside of work. So when I'm baking, maybe I might find 2 or 3 recipes on the internet, but it's hard to know if a recipe on the internet's actually going to work. And so I can use my knowledge of units and measurement, math, to compare them, even if they might look different. I can look at the ratios of all the ingredients and the different techniques and say, well, what is in common between all these recipes and how might that work for me? I also think that STEM is really useful in just dealing with all the information you get in your life. So that might be something on the internet, like news, or maybe an advertisement, or just understanding data, like my solving the water use problems in my garden. It's nice to understand how an experiment works, how a variable works, and the ways that something that might seem intuitive may turn out to not be true because we don't completely understand it. And I think that STEM can really help you make sense of the world around you in your everyday life. It really can just be a way of living and thinking about the world. I really want to thank all of you for watching my video, and if you have a question for me, you can tweet at JGI, and I will respond to your tweet and answer any questions you have. Thank you very much. Bye.